Imagine a breakthrough that could bring new hope to children suffering from Barth Syndrome, a devastating rare genetic disorder. But here's where it gets controversial. Researchers have identified a previously unknown gene that might be the key to unlocking effective treatments for this condition, challenging previous assumptions about the complexity of the disease. Barth syndrome is a rare inherited disorder linked to the X chromosome, caused by mutations in the Tafazzin gene. This gene plays a critical role in maintaining healthy mitochondria, the powerhouses of our cells that generate the energy we need to survive. When the Tafazzin gene is faulty, it leads to a shortage of a vital lipid called Cardiolipin, Cl, and an excessive buildup of a harmful lipid known as Monolysocardiolipin, MLCL. This imbalance disrupts mitochondrial structure and function, impairing the cell's ability to produce energy efficiently. This disorder primarily affects boys and manifests with symptoms such as muscle weakness, increased susceptibility to infections, and a serious heart condition called cardiomyopathy. While heart transplants can offer some relief for cardiac issues, many children afflicted with Barth syndrome unfortunately do not survive beyond early childhood. However, a compelling new study published recently in the prestigious journal Nature might shift that grim outlook. This international research effort involved experts from both academia and industry, including notable institutions like the Hospital for Sick Children and the University of Toronto. Their paper, titled Genetic Suppression Features ABHD18 as a Barth Syndrome Therapeutic Target, reveals a groundbreaking discovery. Inhibiting a gene called ABHD18 can actually restore mitochondrial health and enhance heart function in laboratory models of Barth syndrome. Why is this such a big deal? Before this study, ABHD18 was virtually a mystery, with no known role in human biology. As Sana Massoud, a former PhD candidate in Jason Moffat's lab at the Hospital for Sick Children, explains, their team conducted extensive experiments, screening for genes that interact with Tafazzin, and uncovered that ABHD18 is a crucial regulator of cardiolipin metabolism. Simply put, ABHD18 influences the pathway responsible for producing cardiolipin and converting it into the harmful MLCL lipid. The scientists demonstrated that ABHD18 acts as a suppressor gene, meaning it can partially counteract the effects of the defective Tafazzin gene. Specifically, the paper details how ABHD18 converts cardiolipin into monolysocardiolipin in laboratory settings, and that disabling ABHD18 in cells and mice shifts the lipid balance back toward healthier cardiolipin levels. Directly repairing the Tafazzin gene remains a challenging task, which makes targeting ABHD18 an exciting and more practical therapeutic alternative. Their preclinical findings reinforce this potential. By using a small molecule inhibitor named ABD646 to block ABHD18's activity, they successfully lowered MLCL concentrations in several Barth syndrome models. What's more, this intervention led to noticeably improved mitochondrial function and better heart health in zebrafish and in cells derived from patients. Vincent Bloman, co-lead author and senior director at Scenic Biotech, enthusiastically remarked that this work represents one of the clearest examples of a disease modifier his team has ever encountered. Jason Moffat, also a professor at the University of Toronto, highlighted the hope this research offers, saying, ABHD18 provides a direct route to addressing the fundamental cause of Barth syndrome and brings genuine optimism for affected families. This discovery perfectly illustrates how cutting-edge genetic research can pave the way for innovative treatments, even for some of the rarest and most challenging diseases. But it also raises important questions. Could targeting ABHD18 have unintended effects in the long term? What are the next steps before these findings translate into therapies for patients? We'd love to hear your thoughts. Do you think this approach could revolutionize treatment for Barth syndrome? Or is it too soon to tell? Join the conversation and let us know where you stand.